up in Alaska in the late 90s, and I clearly didn't get the memo that it was supposed to smell bad, but nonetheless was always outdoors, and so it's really exciting that I actually get to get paid to do this kind of work. That leads me to the work that I'm doing now, and like I said, I study different types of pollution that occur in aquatic ecosystems as mixtures, and I try to understand what the effects are of that pollution on the things that are living in the streams. And so most of my work is focused locally in the Humber River, and then also internationally working in the Red River in Vietnam, in Southeast Asia. So I know most of you are motivated by these things: salmon, salmonids, other sport fish, things like that. And so you're probably not going to care as much about what I'm really focusing on today, which is the benthic macroinvertebrates. But I did do some fly tying with Pat Kelly, and I do know that you as fishermen are really interested in what the invertebrates are that are found in these streams and rivers and lakes that you're fishing in. And so my argument here is that you should care about the invertebrates, because not only are they the foods for the fish, but you need to know what invertebrates are found in the rivers and streams, because you need to make your flies look like that so you can catch your fish and continue to enjoy fly fishing. The other thing that I want you to think about is why it's important for us to protect and maintain and monitor what's going on in our freshwater ecosystems. So you can see here that freshwater habitats occupy less than 1% of the Earth's surface, and yet support about 10% of all known species, a third of which are invertebrate species. And we know that climate change and other stressors are impacting freshwater species, and that freshwater is really important to us, not only for our fishing, but also for other reasons, for economic reasons, for ecology, for our health and wellness. And so there's a whole bunch of reasons why it's important for us as community members and people who live and dwell in these areas with fresh water, for us to care about them and protect them. And so when we're thinking about things that stress out fresh water, this was me sort of starting to brainstorm what are the impacts to freshwater ecosystems. And immediately these are the things that came to mind when we have our toilet waste that goes to our wastewater treatments, when we create industrial products, we're releasing pollutants into the water, when we create new cities and build new roads and new buildings, there's lots of waste that ends up in freshwater ecosystems. We change our rivers, we put them underground, we channelize them, we add rocks and cement to the sides, sometimes we get rid of them completely and we fill them in. And this leads to creating new opportunities for things like invasive species and pathogens to enter our ecosystems. So what that means is the biota that are already there, the fish that you care about, and the invertebrates that they eat, are stressed out. And so they're going to respond to that in one way or another. And of course, especially as we've been hearing in the news lately, climate change is sort of added on top of all of those stressors and causing our freshwater ecosystems to be stressed, perhaps more stressed than they've ever been. And so my focus really, like I mentioned in the video, looks at urbanization. I'm interested in how the development of cities, the running of cities, how that contributes stresses or stressors to freshwater ecosystems. And specifically, I'm interested in road pollution or road runoff. So this is a little schematic diagram. You've got the back of your car here, you've got the road and perhaps the sidewalk over here, and I'm interested in what sort of mixtures of contaminants are entering the freshwater ecosystems by way of this road. So if we think about right now, we just had a really heavy snowstorm, the snow plows went through, they got rid of the snow, and what did they put down? They put down road salt, right? And some of you may have heard about this already. We've got really, really high levels of road salt in these urban areas, close to roads, and so that is one of the stressors that I like to study. The other thing is, we've got our tires, that when they rub against the road, they're creating tiny bits of rubber, which I call tire dust, or are commonly referred to as tire wear particles. Of course, our cars are made of metal, and so through time and wear, they release metals onto the road, and also which eventually flush into our freshwater systems, particularly copper and zinc. And lastly, the gasoline that we burn or the diesel fuel that we burn is emitted as exhaust fumes, and that releases a suite of chemicals called polycyclic aromatic hydrocarbons, or PAHs. But I'm going to save you the headache of the chemistry, and I'm not going to talk about metals and PAHs today. I'm going to focus just on road salt and tire dust. So let's talk about road salt. This is a graph that I got from the Credit Valley Conservation. So this information is available to you online. And Credit Valley Conservation, along with the Toronto and Region Conservation Authority, and I'm sure many other conservation authorities that I haven't looked into yet, have these sorts of biomonitoring programs in place, where they stick some loggers into the streams that we're interested in studying, and those loggers will monitor things like temperature, things like salinity or how salty a stream is, other things like metals, oxygen, we were talking about anoxia earlier, because they're interested in what's changing in these streams over time. And I don't want you to focus too much on what you're looking at, but basically we have December over here, and we have March over here, and this is just one creek that the Credit Valley Conservation monitors, and what you can see is that over the course of the winter we have these peaks. And those are peaks in chloride or salt. So salt is sodium chloride, so we're talking about the chloride ion, and we're seeing pulses in salt. And that's probably happening when it snows, we add salt to the road, and then all of that snow melts and it enters our freshwater systems. And so not only are we seeing these pulses over the course of the winter that inherently our fish and our invertebrates are going to be exposed to, but we're also seeing that over 10-year, 5-year periods of time, the baseline levels of salt in these freshwater systems are increasing quite drastically. And this is, again, not just in the Humber. We're seeing it in Credit Valley. We're seeing it in the Muskokas. We're seeing it around Barrie, pretty much anywhere in southern Ontario where we use salt and where there is some degree of urbanization. So if we look at this map by the TRCA, this Toronto proper is around here, and this goes out to about Etobicoke area and out to about Brothers, Duffins Creek area, if you're familiar. And what we can see is that the size of the dot, all these dots on the map, and the color of the dot corresponds to how salty it is. So the smaller it is and the greener it is, the less salt there is. And the more red and dark red it is and the larger it is, the saltier it is. And what's unsurprising is that closer to the center of the city, you have more red, and as you move out, you have less red. But what's perhaps more interesting is that we have a major highway here and a major highway here. And all of our red spots, despite being quite far out of the city, have got these really large red dots, which suggests that roads and these impervious surfaces, things like asphalt, might be contributing sources of salt to our freshwater ecosystems. And so seeing this, A, was shocking for me, and B, was what really motivated my work. And so I conducted some field work a year ago now, where I went into the streams and I was trying to get a better sense of what was going on in the Humber River over the course of the winter. And so this is what I found. I had 3 sites that I sampled in. I put in those loggers, dumped them in the stream. Well, didn't quite dump them, but put them in the stream. They lived there for about 5 months. And every 15 minutes they recorded the salinity and the temperature. So what I'm showing you here, orange is my most urban site, that's the top line. Green is a suburban site, that's in Brampton. And blue is a rural site, that was up in Albion Hills Conservation Area, close to Caledon. And what you can see is all of these sites have those peaks that I showed you that Credit Valley was seeing. And the peaks and the range of saltiness or salinity in the urban area is much, much more drastic than it is in the suburban area and the rural area. And so what we saw in Credit Valley Conservation and what we were seeing over these 5-10-year periods in other studies was in fact happening in the Humber River. And so the question becomes, okay, we know that the salt is changing. We know that there's greater salt concentrations in the more urban areas and less fluctuations in the more rural areas. But the question is, what's happening to the things that we care about? What's happening to these fish? What's happening to these invertebrates? Photographs that I took with my microscope. So these are some of the invertebrates that I found in my sites, and some of them might be familiar to you, or am I Yes? No? Okay. And some of these are the ones that we care about. Some of these are the ones that we're like, yeah, we're happy to see them. And some of them, the ones are ones that we're not so happy to see. And what I was doing by looking at these invertebrates was trying to gauge, based on what invertebrates I was finding or not finding, have that be an indicator of how healthy the stream was. So I'm sure, as you know, as fishermen, the fish like mayflies, stoneflies, caddisflies, am I right? Is there anything else? No, mostly those ones, right?
Terrestrial.
Terrestrial insects. So as an aquatic ecologist, I'm focusing on those larval phases of insects, the stoneflies, caddisflies, mayflies. And as a scientist, I know that those are the 3 species that are indicator species. They're the ones that are sensitive to things like pollution, things like low oxygen levels. So you as fishermen know if those things aren't there, you're not going to find fish. I know if those things aren't there, there's something going on in that stream. And so that's the basic idea here, is that I'm looking at what species I'm finding and how many species I'm finding to tell me about how healthy the stream is. I just thought I would put this video in here. I don't know if it'll run, but this is a little video of some mayflies that we have in the lab. These are test species that we culture, and so we grow them in the lab, and then we'll expose them to different contaminants to try and see how well they survive or how poorly they survive, or if their behavior changes. And I just thought their gills were beautiful. I always see these bugs when they're dead, and so it was nice to see them. And so this is basically the question that I'm trying to answer in this part of my research. How are urban streams, particularly the Humber River, impacted by road runoff? And so what I did to answer this question was I went into the field. And as someone who did lots of camping as a kid, it was basically like getting paid to go on a camping trip. And so I loaded up my stuff in our field van at school. I had 2 assistants. We go into the stream, and mind you, road salt's applied in the winter, and so I'm doing my sampling in the winter. Basically a year from now, I had done a field day. So I'm in the stream, the temperature is between 0 and 4 degrees. I'm wearing long johns, I'm wearing sweatpants, I'm wearing neoprene waders, I have my Under Armour top, my fleece sweater, my jacket, my head buff, and then my hat, and then my hood on top. I have hand warmers, I have foot warmers, and we have hot water in my water bottle that every time my hands freeze, we pour hot water on my hands. And that's how fieldwork goes when you're doing it in the winter. So here is a picture of me collecting the invertebrates. We use what's called a Hess sampler. It looks kind of like a giant soup stock pot that doesn't have a bottom. So you smush it down into the stream so it gets some of the sediment in. And then there's a big net that goes through your legs, and the stream is coming forward and it's rushing through a little net at the front of the Hess sampler. And then put your hands inside, and actually I think I'll be showing you a video of it later, but you put your hands inside and you clean off all the rocks. As you know, the invertebrates cling to the bottoms of the rocks. They'll burrow in the sediment a little bit, and so you're basically rustling up the sediment, dislodging these invertebrates, and then they're going to flow into the net behind you. And then that becomes your sample. You pour it into a sample jar. You add some ethanol, which will kill them and preserve them, and that's what I take home and I'm counting in the lab. I just told you this whole slide. So we collect our invertebrates. There's the net there, and so it's in between my legs here. And then we take it back on land, and the bottom part, the little canister, unscrews, and that gets dumped into a container, and we top it up with ethanol. That preserves them like you would see a specimen at the museum. And then we take it back to the lab. This isn't me, but we've got tons of undergrads that help out with this research, and we count the invertebrates. So for me, I'm an amateur invertebrate taxonomist, so I'm identifying them just to a pretty coarse taxonomic resolution. Things like, is it a midge, is it a mayfly, is it a stonefly, is it a caddisfly, is it a bivalve, is it a snail? Nothing more specific than that. I might have to learn to be better than that, but for now that's what I'm doing. And so
there's some books back there.
I probably
entomology
have them.
Books.
I should. I've got. I call it the Bible. It's about this big and this wide, and I've still got to read it.
It's really hard to read.
Yeah, it's an art. The same way you learn how to identify these fish, there's probably 2 to 3 times more, if not more, diversity of invertebrates, and so You obviously see the common ones just like you do with the fish, but it's an art, and it's a dying art as well, because we've got DNA to identify these species, or we've got DNA techniques to identify these species. So the value of taxonomists and naturalists is slowly decreasing. I would argue that they're still really important, but it's less valued, I would say, in the academic community. Okay, so this is me showing you some examples of some of the data that I have, and I'll just let you know that this is a work in progress. I'm in my second year of 5 of my PhD, and so I'm still chugging along pretty much every day in the lab, picking through my samples. And so what I'm showing you today is likely going to change once I've counted all of my samples, but we are seeing some pretty interesting things, so I thought I would show them to you. So like I said, I've got the 3 sites that I sampled in, and I looked at how they were changing in space and also in time. And so I have 1 site here that I counted in my urban site in April, and 2 sites in my suburban site, that place in Brampton, in April and in November of the previous year. And what I want you really to look at here is how many different color boxes there are in each bar. And so what to me becomes very obvious, especially between this one and the one on your far right, is that the one on the far right has got a lot more boxes. It's got a lot more taxa. It's a lot more diverse. So you've got a lot more taxa or species or groups of invertebrates in this site, in your suburban site, compared to your urban site. And particularly those 3 taxa that we're concerned about, mayflies, stoneflies, caddisflies, that the fish like to eat, those are found in our suburban sites with the stars, and they're not in our urban site at all. We can't find them in my site in Toronto, but we can find them in Brampton. And so that to me suggests that if you were fishing, you'd be more likely to find a fish in the suburban site in Brampton than you would in Toronto. Okay, so what we're seeing when it comes to our invertebrates is that as you move from inside of the city to out of the city, you go from none of those important species, our stoneflies, caddisflies, and mayflies. And the question is, and our fish are probably more likely to be found in those rural areas. Like I said, it's possible that this will change as I add more data and as I continue counting, but I guess stay tuned, and maybe I'll come back in another 5 years and have a story. And so the other thing that I'm looking at is microplastics in the Humber River. And I thought I would talk about this because it's a new and growing field. Most of the people in my lab study microplastics, and so I think I'm edged towards talking about it as well, because that's what everyone else does in my lab. But also because we know a lot more about salt, and we have a lot more sort of policies and management practices already in place to deal with salt and the salt issue that we know a lot about than we do with plastics and microplastics. And so I thought it was interesting to look at this contrast and to see how the trends are similar and different. So I was mostly interested, like I said, in those tiny bits of rubber that are created when the tires rub against the road. So like I said, I call that tire dust. It's made of rubber, which you can see here in the gray. The colors aren't great, but you'll have to trust me on that. But when we're counting microplastics, we also look for we count all of the microplastics that we see in a particular sample. And when we're counting microplastics, we identify them in similar ways to how we would identify invertebrates. We almost have like a taxonomy of microplastics. So we organize them based on their shape, based on their color, based on their size. And all I want to point out in this figure here, again, we have urban here, suburban here, and rural here. So there's a little bit of variability within sites when you sample on certain sampling days. But what you can see is there is more microplastics in general in our urban site compared to our suburban and rural. And when we're looking just at the rubber, which is what I've circled here in the gray, we've got about 50% of our samples in our urban areas are comprised of rubber, whereas in our suburban and rural sites, there's significantly less rubber. Now, one of the other reasons why I think microplastics are interesting is, and one of the things that I found the most interesting about them, is that they can basically act as sponges for other contaminants. So if I have PAHs, which I'm studying, PAHs in the environment, say there's been an oil leak or something like that, or I'm close to the oil sands. I know we're here in Ontario, so we're not, but say there are PAHs in the environment, and here comes a little plastic floating in the river. Those PAHs are going to stick to the microplastic. The microplastic that's floating around has these PAHs stuck to it, and now your fish confuses this piece of plastic for a piece of food, and so it eats that plastic. Now not only has that fish eaten plastic, but it's also consumed those chemicals as well, which we know some of these PAHs are carcinogens. And so it's going to be affected not only by the plastic, but also the chemicals. And so I find it interesting, unsurprising, but nonetheless interesting that we're finding these higher concentrations of microplastics in our urban sites. But we're still finding microplastics in our rural sites that have noticeably less human impact. I mean, it's in a conservation area. There's one gravel road that is in this area, and it's up basically in the headwaters. So that was very interesting for me. So when we're looking overall, as we move from in the city to out, we're seeing more microplastics in the urban areas and less microplastics in the more rural areas. And again, I do expect that these trends might change as I continue processing more samples. I thought about, since I was talking about salt and I was talking about microplastics, I want you to leave here not thinking, oh my gosh, we're doomed, the Humber River is so dirty, there's nothing I can do about it. So know that there are lots of things that you can do both as a community group and as individuals in your home, work, and other life. So the first thing is to shovel first. Oftentimes we're lazy, we don't shovel the snow. Get rid of the snow with your shovels, and that's going to reduce the amount of salt you need to use, if any at all. Also, if everyone used snow tires and wore proper footwear, we would be less concerned about safety. So I would encourage you to get your snow tires, use your snow tires, encourage others to do the same. When you're using salt, you probably are using too much, and so this is what the Ministry of Transport recommends. So we're still meeting safety standards if you're seeing this much salt per unit area, and this is what we tend to see, or more. Sometimes we see like an inch of salt. There was recently a pilot study done at Ryerson University where half of campus they salted as usual, and the other half of campus they brined. So you mix the salt with water and then you apply it as a brine. That brine was equally effective to the crystals, and it used less salt. So that is one way that private businesses could A, reduce their costs, and B, maintain a high level of safety. And someone mentioned plants as well. Avoid applying salt to plants. You might see these brown patches on the sidewalks. That's probably salt damage to the plants. And the last thing I would mention is when thinking about your cars and reducing salt on your own cars, skip the wash and save the lake. So try and wash your cars less. I know it means damaging your cars a little bit more, but it also means flushing less of that into the water. When thinking about microplastics, pretty much all of the microplastics that we see in the environment are coming from macroplastics, or regular sized plastics. And so one of the best things you can do to reduce microplastics in the environment is to remove litter, trash, and macroplastics from the environment. And so one of the things that I do with an outreach team that I'm a part of at the university called the U of T Trash Team is we lead cleanups. So we get community members together. We've done this all over the city and all over the world, where we get community groups together, you get some gloves, you get recycling bags and trash bags, and you pick up trash in your community, by your river, at your waterfront. Ultimately, all of the trash that we have on land can and might end up in our freshwater ecosystems, either by rain or by wind. And so if you're able to remove that and prevent it from entering our environment, then we can prevent it from becoming a microplastic. Some other suggestion that I have said is share what you know with others. So you're welcome to come and talk to me about what we talked about today, or if you have any other questions. Walk or bike to school to reduce the amount you're actually using your car and generating tire dust. And if you're feeling very passionately about this, especially as a group that cares about fish, let local government and decision makers know that this is an issue that you care about. And it might be something as simple as writing a letter saying, hey, we learned about road salt, or we learned about microplastics, or we learned about both. This is an issue we care about. We want to see sport fish in our rivers and streams, and we'd like for you to take action on it. It doesn't even have to have action steps, but just the fact, usually when a decision maker or government official sees a letter, one letter tends to represent the opinion of 1,000 people. And so writing that one letter can actually really help to amplify your own voices and the voices of others. And like I said, reduce your salt use, which I talked about earlier, in your neighborhood, in your home, and at your school. So, with that, I will leave this on here for questions. But if you don't have an opportunity to chat with me after this, please feel free to get in touch. And thank you for having me.